So last speaker of the day is Sébastien Ballesta, who is now maître de conférence at University of Strasbourg. But he will talk about his postdoc work, I think.
You're well informed, Thomas. So hello, everybody. Thank you. I would like to thank the SBDM organizer. I mean, the first time for me coming to this great conference and first time talking. And yeah, as Thomas said, I will present you the result from my postdoc that I did in WashU in the team of Camilo Padua-Scopa. So I'm going to talk about the contribution, the causal contribution of orbitofrontal cortex in value-based economic decision. Ça marche ça? Ouais, ça marche. Okay, so let's define some economic decision. So it's when you need to select amongst more than one option. So we have at least two available options. The options are value amongst more than one dimension. For instance, if I ask you if you prefer $10 or $20, it's not an economic decision, even so it's with money. And there is no intrinsically correct answer, meaning it's based on your subjective preference. So if you, the typical situation where people are doing economic decision is where you after the conference you go with your colleagues, which restaurant to choose when you go inside the restaurant, what to order, and arguably at the end of the meal, who is going to pay the bill. That can be considered as an economic decision. So the idea is to find the biological basis of this decision, and we are studying that in monkeys so that we can study the brain in an invasive manner. So obviously, monkeys are not going to restaurants, but we kind of like put the monkey kind of in front of the bar a little bit. Because he has the choice, so this is a typical task that Camilo have developed during his postdoc in 2006. So I'm going to summarize like 20 years of almost 20 years of research to come to the macro-stimulation experiment that I did with Camilo. So with this task, you have the monkey is facing a screen. You need to fixate this little dot. And you have the option, the choice between two offers. The type of the offer is informed by the color of the square, and the number of drops that is going to have is based on the number of square. So here we got like peppermint tea and apple juice. So A will always be the most preferred juice. After here on the classical task, the monkey does a fixation dot dim, and then the monkey can decide here he prefer apple juice. So in this example, you can measure the subjective preference of the monkey. It's based on this decision, and if you change the amount of A and B that you propose to the monkey, then the big dots are the decision of the monkey in these different conditions. And you see that here he was mostly choosing A, here mostly choosing, like always choosing B. And here we have like some split decision where the monkey is not always choosing the same thing. And this is the interesting part of the behavior, because thanks to that, we can calculate the relative value. And here we can, by fitting a sigmoid and looking at the indifference point, or the point at 50%. Going back on this scale, you can say that here the monkey, for the monkey, 1 drop of juice equal to 4.1 drop of peppermint tea. So Camilo have studied this in the orbitofrontal cortex. So why is the orbitofrontal cortex interesting for economic choice? It's because this area is particularly well connected upstream of the decision, so all the sensory and limbic area. And downstream on the decision when you need to do an action like a premotor, for instance. The other interesting part is that the OFC is representing the value of the offer. So it's not at all the only part of the brain doing so, but it's the part of the brain that is doing that in the most independently from spatial organization. So whether the offers are on the left, right, in front, or behind you, for the OFC, it doesn't matter in somehow, and which get the flavor of the fact that in OFC, some abstract things are going on. Through his study, he built this working hypothesis that within the OFC, there is this neural network that some of you may know. Okay. So you got the inputs, the offer value, and the output, the chosen value and chosen juice. And I'm going to describe a little bit this network for those that are not okay with it. Okay, so first, the offer value. So on this graph, you have like, again, the big dots are the monkey's decision. Here, this is a choice matrix. And on the top, you have the firing profile of a single cell here according to the decision type. So you see for these neurons, as you're increasing the quantity of B, and the firing rate is also increasing. But if you're increasing the quantity of A, nothing happened. So it would be an offer B neurons that is specific for the value of the less preferred juice. And here would be the activity profile of the population of the offer B neurons. So these neurons, you have the same for the offer A, and these neurons are connected, so supposed to be connected to others that are called the chosen juice here. And so these neurons are not sensitive to value, they are not encoding value, but they are encoding the identity of the offer. So here, if the monkey have chosen B, it would A, sorry, it would be high, and if he has not chosen A, it would be low. So it's like 0 and 1 neuron saying like you have chosen A or not. And the last one are the chosen value. So they are not sensitive to identity, but they are sensitive to value, independent of the identity of what you. This is really the value of what you have in front of you. So this neural network is interesting because we are working hypothesis is that the decision is taken by this neural network in a way that the network can oscillate between some state that you have this chosen A that are active or chosen B, and the one that managed to be active, it would predict the one that is going to be chosen by the monkey. So it's a neural network that oscillate between different states that represent choice option. It's a little bit like in the movie from Terry Gilliam. So I'm not the first one to use this metaphor. I have borrowed it from Thomas Boraud. And so in this dystopian movie, you have people in an office, and in his manual, he says that when you need to make a decision, you need to ask the executive decision maker, which is this device that is provided by the office. And so you load the device, and after you ask, you get a question, it's going to answer to you whether it's like yes or no at 50%. Obviously, here it's no. Why the main character needed a yes. So the idea is that maybe this neural network can work a little bit like that, with the idea of having maybe a bias on the initial condition. This bias actually can be found inside the chosen juice neuron. This is something that Camilo have described and have called choice hysteresis, where so when the monkey have a bias to choose the last chosen option. So if he's chosen A on the last trial, he's more likely to choose A on next trial, same with B. So this is a difference in behavior. And actually, this bias can be read out in the activity of the baseline activity. So before that you present the offer, this bias can be read out in the activity of the chosen juice cell, which is a good, good information, the good sign that maybe there is a causal link between the activity of this neuron and the choice of the monkey. And the idea again would be like this baseline activity would be as you bias this little thing here, and so you bias for the juice that you have chosen before. So my duty in Camilo's lab was to pass from a simultaneous economic decision to sequential economic decision. So at the beginning it seems like it's a super incremental little tiny changes in the behavior in the task, but it allows for different things that were really important at this time. So you can dissociate the evaluation process from the comparison process from the action process. So here to do so here because we show offer 1 first, then inter offer 2 is waiting, and then is doing the saccade according to the color of the target that you want to choose, and the dots were randomly assigned on the side. So that the monkey cannot like predict that I'm going to choose B, so that I'm going to do my saccade on the left part. He was not able to do so. The other important thing that we realize that if you are not constraining well your matrix of choice, that is not possible because the monkey may predict the value of offer 2 just by seeing offer 1. And to prevent that, you have to construct a matrix of choice like that. So it's each point is a combination of offers, offer A and offer B. And so you need, like, for instance, when if the monkey was seeing 2A on the screen on the offer 1, he cannot predict the second one because he can either have 1B, 3B, 5B, or 8B. He cannot know which one is going to happen and also which one is going to choose. But to do so, you need to predict the relative value, and so you need to become a monkey's barista. So that because you need to make sure that the monkey for this day maybe is like this one like a cherry juice, and he's not really into salty water or stuff like that. And he can because you need to predict where this indifference line is going to be. So sometimes you can put the juice. The monkey, you realize that for some reason he's fed up with apple juice today, and so you just empty everything and go back. And especially disturbing when you have a nice neuron, but anyway. So the task design prevents the prediction of offer 2 value based only on offer 1. It seems pointless, but it was crucial. By recording neurons in this task, we found something that was really interesting and was consistent with previous finding. So this is a chosen juice neuron. So again, a neuron that is not supposed to be sensitive to value. Is a 0 and 1 neuron for do you have like A or B in front of you. This was a trial where you have first the non-encoded juice by the neuron, and on the offer 2, the encoded juice by the neuron. And in this neuron, we can find back a value signal of the other type of juice. So which somehow was strange because these neurons were not supposed to encode value, and you see this value was negatively encoded, so it was inhibited by the value of the other offer. So this baseline thing is really recall us the choice hysteresis story. So it would be like kind of a biasing the decision here, and this may be a neural mechanism where that allows the circuit to make the decision, and so we call it circuit inhibition. But the interesting, the puzzling part was within the OFC, we were not able to find a recall of the offer value. And not even like on the so we wonder this signal here, which again corresponds to the value of this offer that was here. Maybe it's coming from another part of the brain, or maybe it's stored within the OFC in a way that is independent from spikes, for instance. There are several people that are working on a synaptic weight or things like that, things that I don't fully understand. But that may be some hypothesis for like how value is memorized and stored between the two offers within OFC or in other part of the brain. So this neural network is not the only one that has been proposed in the past to take a decision in a frontal cortex. There was also this more simple hypothesis of mutual inhibition, where you got like the offer value is inhibiting the other offer value, and so that one of the two is winning because you cannot like inhibit each other indefinitely. So thanks to our data, we can have some prediction about like if the network was wired like that, you should, for instance, so this is like an offer value cell. So when the encoded juice was presented first or second, and since the monkey cannot forecast the value of the second offer, if we had like a mutual inhibition like that, we should see like a decrease in activity when the offer was presented first or second, because on second it should be inhibited by the other, which we are not seeing, and also we are not finding back the negative signal of the value of this offer here. So for us, in our hands, there is no encoding of the value of offer 1 during offer 2 time windows in this neural type. And also, we did not find any cells that encode significantly the value of offer 1 minus offer 2. And the whole story of the mutual inhibition was based on the results published in 2014, where using like another way to analyze the neuron based more on GLM, they were finding that during offer 2 time windows, there was this anti-correlation between the value of offer 1 and value of offer 2. So we were not like really confident about this result, and so we tried to reproduce it. we managed to reproduce it. But we realized that if you take, so Jean was talking about range adaptation. I will talk more about that. But if we take neurons in session where like you have like unequal value range, you have that. And if you take session where it's like equal value range, this correlation is totally disappearing. And it's explained by a really kind of like simple thing in SOMO. If you take the activity profile of like here, it's a chosen juice, which you see like it's a A, B, or BA trials on offer 2 time window. So it's the neuron is high when he is in front of B, and it's low when he's in front of A. And so this is the model that is best explaining the activity of the neuron. Now, if you take this neuron in a session where you have different range of value, you see here the value of A would be from 0 to 5 and value of B from 0 to almost 15. And you do the regression, you will have, because of this difference in range, a correlation that is positive here, negative here, and even a correlation with value difference. And this, having that, so as soon as you get like neuron with label line, that neurons that are sensitive to A or B, and a difference in range, in value of range, you will have this this type of result. So yeah, just to, you know, because it was buried in the supplementary of this paper, but for us, it was really important to show that this way of thinking the neural network within OFC was not the way that we were agreeing on. So today's question now. So at this point, there was a lot of remaining question about like OFC and decision making. First, isn't because you get a value signal that it's causally related to the decision. For instance, if you look at EMG signal, if I ask you to grab, I don't know, like a bottle of Coca-Cola or a piece of gold, you will be faster to grab the bottle full of gold, and it's activity of your muscle, and you're arguably not taking decision with your muscle. So it's not because you got value signals that it's causally implicated into decision. So the first thing to do was that we want to show is that the value signal are causally involved into economic decision. The second task would be to test whether is there a value comparison process that take place within the OFC. And to do so, we did like a micro simulation study in OFC during economic decisions. So the main paper was published in Nature in 2020, and the follow-up paper in 2022 that I'm going to present as well today. So the task is the same that I presented lately. The sequential offer, the first A, second B. Here, and so we stimulate offer during offer 1. And we have this, so this is the behavior of the monkey again, so the decision. In light, you get when stimulation is off, when in dark, when stimulation is on. In reddish, A, B trial, so A first, B second, and in bluish, B first, A second. We can look at 3 things on this sigmoid. First is the steepness of the sigmoid. That is going to be informative of the choice variability, whether the decision is more noisier or not when we stimulate. So during offer 1, there was no change in choice variability. The relative value of AAB, you know, maybe for some reason, maybe the stimulation would increase the value of like the most preferred juice and not the other one. And the distance that you get between the steam on and the steam off in AB and BA, which is going to be informative whether you're going to increase or decrease the value of the offer that you are stimulating. And here during offer 1, by doing the stimulation, we were decreasing the value of the stimulated offer, which I have to say was going against our prediction, but you know, whatever. So during offer 2, we have the same effect than offer 1, reverse because you know you stimulate offer 2, but we also add an effect on the steepness. Okay, which was informative of the fact that we were disrupting the decision when we stimulate during offer 2. So this was like one example and this is the population result. stimulating during offer 1. So if you have the ellipse, which is like on the line, it's not significant. And if it's like a little bit displaced, it means that you have an effect, a significant effect. So we have on the population level, we have an effect on the order bias. When stimulating on offer 2, when we stimulating on offer 1, sorry, offer 2 time window, we had again the effect on the order bias, but on the other side, and we had the effect on the choice variability. So we were able to both disrupt the evaluation process, but also the comparison process. Okay. My God. Okay, so thanks to Jean Denisot, I may skip this slide because you all know now about range adaptation. And so the neurons need to adapt because they have a physiological limits in their activity. And when you increase the value in the environment, the meaning of an action potential is changing in terms of the meaning in terms of value. And in the context of microstimulation, this was really interesting because we can make some prediction. If the meaning of an action potential is changing in terms of value because of the range adaptation, when you microstimulate, you arguably increase the activity of the neuron. And if you increase the activity of the neuron of the same level of firing rate, but the meaning in terms of value is changing because of difference in range that you propose to the monkey for the A, for B or A, it means that you will give an advantages to the offer that have the highest value range. So it was a little treat. And when we test this hypothesis, actually we found exactly what was predicted. This is the difference in value range between A and B, and this is the difference in the relative value on stim on and stim off. And you have this regression independently for the 2 monkeys, and even for the third one, because in another data set we were able to reproduce it. You have this regression. So before I show you that there was no effect on the relative value, but because it was a mean of that. A mean of this dimension, but if you consider it according to the difference in the relative value, you will find that, because again, according to our prediction, when you increase the firing rate, it gives an advantages to the neurons that were 1 spike per second have the highest definition in value. So I didn't mention that anything about current intensity, but because I was kind of exploring the thing, nobody had done OFC stimulation before, and in the frontal cortex is kind of tricky. The stimulation you don't really know what's going to happen. So I tried a lot of different current intensity, and we have some effects that are here on some current intensity or not. So at really high current, we have the choice variability effect only during offer 2, and the order bias, but not the range dependent bias. That is the effect that I just showed you the slide before. When you decrease the intensity, you don't have the choice variability anymore. You have a lower effect on the order bias, and you start to have the range dependent bias. When you decrease again, you don't have all of these effects that are all disappearing. You only have the range dependent bias. And now, guess what? When you decrease even more the stimulation, you would expect to have no effect at all. And out of my great surprise, when you stimulate by really low current, this is the highest disruption that we can do to the network. So it was real puzzling. I have to say, that I did this observation myself when I was exploring the thing. And it was hard to believe, but it was also reproduced by another student on another monkey. So we say, okay, it's real, and we decided to publish it. And so we are not fully understanding what's going on, but we have some hypotheses about what's going on. So first, let's go back on the what microstimulation are doing to neurons. Because with optogenetic and dreads, people say things that microstimulation are not fancy anymore. But I hope that I can convince you that maybe it's a technique that it's worth keeping on doing. So microstimulation. So when you stimulate at a high level, high intensity, you are messing up with everything. You are activating the whole neurons. You can go do action potential in both directions. you are activating the fiber of passage, arguably. And you really activate the network in a really disruptive way, so that some people are talking about neuronal hijacking, because the output of the area that you are stimulating is going to be the train of stimulation that you're going to do. So it's really like you hijack the whole area, and you are not taking into account the balance between activation and inhibition within the network. While when you are doing, when you're going down in intensity, you're doing like a transsynaptic physiological activation, because actually you are not triggering action potential directly, but you are triggering the most sensitive to current phenomena in neurons, which are the exocytosis of the neurotransmitter. They are going to activate the other neurons, so you're doing a more physiological activation. And now when you're going down, we don't know, but we do have a hypothesis, two lines of hypothesis. That one would be that it's spike related, meaning that because you are really close to the limit of triggering a spike inside this neuron in the literature. So it's hard to say, are we triggering a spike or not when stimulating at this really low current? But if we consider that we were triggering spikes, so we can maybe say, okay, maybe we were activating some cell in the network, but not the other. And maybe that was the interneurons, and maybe chosen value are mostly interneurons. Yeah, it's a lot of maybe. But maybe this activation, that specific activation of chosen value cell may disrupt what I call circuit inhibition before. And by pointing to this specific cell type, cell function, this is why we were disrupting the decision only and not the valuation. The other hypothesis would be spike unrelated. You know, when you have an anomaly, this is where things can be fun, and you can be creative about what is displaying your result. Maybe there is something that we are disrupting at the synaptic level. Maybe it's about the ion flow. There are like some obscure literature on the theoretical effects of microstimulation on the retina that say that at this level, you are just inversing the ion flow of sodium or calcium. Maybe it has to do with the activation of astrocyte compared to the balance of activation astrocyte neurons. I don't know, and I will surely not have the answer. Because we need to have an experiment at the degree of details that is not able easily to do with monkeys. But still, even if we don't have the effect, the explanation of the effect, we can say that here this is this slide, that value signal with OFC are causally linked to primates' economic decision, and thanks to the fact that we are disrupting the decision using really low intensity, and so no fiber of passage, we can say that value are locally compared within a neural network that produce circuit inhibition. But okay, so really fast, I would say that this is super interesting, but I would not argue, that we understand everything about decision by understanding, all these little things, because OFC is maybe the size of my little finger in the monkey's head, so it's more at play than that. And maybe some other way of like more broader way of understanding decision, like for instance what Paul Sisek is doing. Can study decision also in a more evolutionary and different part of the brain. And actually, what I've shown you would fit into only this part of this whole thing. And as Thomas said, now I'm studying decision in a more naturalistic environment in Strasbourg, where we have monkeys in semi-free ranging settings, and we can study decision using these devices, and we can also have a decision in an economic and a social way, and socio-economic decision as a paper that, and I'm going to thank you and thank especially so Camilo, Wei Kang, that I've worked on this paper with. He was a PhD, and Heidi, the name is I cannot pronounce it, but Schnocht. But Heidi was a lab manager, and she was part of the success, and the monkey, and you for your attention. Thank you.
Thank you for the great talk. I had a question about your on the slide where you showed the microstimulations, if you go just a little bit higher in terms of stimulation, you have the opposite effect of the, over there. So it's going down, well, in the opposite direction, in the with the red bars. Do you have an explanation of why it's going in the opposite direction?
Yeah, this one is not significant, so you know, I can say that. But you know, like you got the little star on the top. But I agree that it's kind of disturbing.
Yeah.
And you know, maybe at some point we may be able to make decision less noisier in some session, but the thing is that it was hard to be because the choice variability is about the steepness of the curve, and to have a good steepness you need to have at least 2 points that are in the indifference that are split choices. And you know, because yeah, it's hard to forecast. We want to have as a less choice possibility to have more data and do more sessions on every day. And so maybe I should maybe have put more offers here to have a better estimation of the steepness. And so maybe here it's about like some you know, outliers or something like that that can have driven this effect. But anyhow, these 2 effects were not significant. So that's, you know, the short answer to that.
Thanks.
I have a question about the intensity of the current. When I was doing stimulations a long time ago, there was this idea that the current intensity was related to the volume of tissue that was affected by the stimulation. So I was wondering whether the proportion of OFC neurons affected by the stimulation and as a function of the underlying interaction that they have in order to compute value and decision could be a factor explaining this.
It cannot be like the only factor. If you try to think about like it's because you are stimulating more OFC, you shouldn't have like something like you see like the range dependent bias. You see like this U-shape effect, and that cannot be explained by only, you know, because if it was only that, we would have like more and more. So because, you know, like you can also point out that we have an effect that is decreasing the value and an effect that is increasing the value. So it's like the effect is kind of complex inside, but for since this one is dependent on range and the other one not, it kind of makes sense. You can have like something go down, and you know, I have no explanation of like why value is decreased when we stimulate the OFC. I guess we are messing with something that we don't know what. But with this spread in volume, the thing is that if you have an effect that is dependent on the current, you can argue that you can have an effect on the center, but have a different effect on the edge. So you know, since we are totally blind about you know what's going on, I had some session where I had another electrode recording 1 millimeter away. So you know, I had some stuff, but we did not publish this. Maybe one day. But it would be super interesting to be able to do stimulation and recording at the same time. It's possible. People have done it in the past. But yeah, it's not going to be me that's going to do that soon. So you know, anybody that is up to that.
Thank you. I think we run out of time. So I just want to thank again all the speakers for this very dense and nice session.